I'm very, very happy to be joined today by Dr. James Kirkland for this week's meeting of the Foresight Health Extension Strategy Group, sponsored by 100 Plus Capital. Today's topic will be studies of aging interventions, and it's fantastic to be joined by Dr. Kirkland. Today's topic is really aligned with work that he has recently been really focused on, which is really conducting or beginning over 12 FDA-regulated studies of senolytics and other agents that target fundamental aging mechanisms. And so this is a really lovely opportunity to discuss potential FDA regulatory questions in this group. So thank you so much for joining. I will share a more extensive bio of you here in the chat, just to highlight perhaps a few things. Dr. James Kirkland is the director of the Robert Arlene Kogod Center on Aging at the Mayo Clinic and Noaber Foundation Professor of Aging Research. He's the principal investigator of the Translational Neuroscience Network, which brings together 8 academic institutions to translate health span interventions from bench to bedside. You're also the president-elect of the National Advisory Council on Aging of the National Institutes of Health, a past chair of the Biological Science section of the Gerontological Society of America, and a member of the Clinical Trials Advisory Panel of the National Institutes of Aging. So there's a lot of expertise, I think, combined in you as one person here. So thank you so much for joining. I will collect questions during your presentations in the chat, and I'm terribly excited for the discussion afterwards. Thank you very much, and yes, the stage is yours.
Thanks. So thanks for the kind introduction. I'll just start by saying I haven't been to one of these meetings before, so I'm not quite sure how things go. One other important thing that I still remain to do in my life, and that's to meet Nir Barzilai, who I see on the screen. And I'm really eager one of these days to meet with him.
It will be my most exciting day. I've been waiting for it for decades. Thank you so much.
Same very much here. So I'll try to share my screen. I'm technologically very inept. So we'll see if this works.
Perfect.
Let me just get this. So is that working all right?
Looks great to me.
Okay. So I'm going to talk a bit about senolytics and sort of the paths we've taken. I'll start off by saying I'm no expert about regulatory steps and hurdles. And I even misspelled steps. It should have said steps, not step. And so I'm just going to go through some of the things that we've done to get ourselves to the point of clinical trials. And there's no right or wrong way of doing things. Just some of these points that I'll make are opinions and things that we found work along the way by trial and error. So I think everybody on the call is aware of the geroscience hypothesis first put forward by Gordon Lithgow, and then people like Felipe Sierra and Ron Kohansky, who I think is on the call, and many others have really developed widespread interest in testing this hypothesis, and that's that fundamental aging mechanisms may be root cause contributors to the geriatric syndromes, chronic diseases, as well as decreased resilience. So our way of approaching developing agents has been to focus on targeting some of the downstream effects that might be contributed to by fundamental aging mechanisms that could be root cause contributors to these conditions. Instead of going after things like lifespan itself, we've gone after age-related disorders where there appears to be at least a reasonable link to fundamental aging processes, and to looking at several of these kinds of conditions in parallel. So it's a different approach than, for example, what's being planned with the TAME trial, where where a single large study would be done. These are multiple small kinds of trials. So we're also interested in testing what we call a unitary hypothesis of fundamental aging mechanisms, and that's that they may all be linked. And it's looking increasingly like if you target one fundamental aging process, you tend to affect the rest. And you tend in animal and cell culture models to impact multiple downstream conditions, as you'd expect if they're a fundamental upstream root cause contributor to these things. So I like to divide fundamental aging processes into 4 categories. And these processes operate not only at sites of phenotypic changes in older individuals, but even in younger individuals, even in children, at sites of onset or etiology of multiple chronic conditions and diseases. And these processes include, and people divide them up into 4 or 7 or 9 or 13 or whatever, but I think they're all much more closely linked than we've previously recognized, if this unitary hypothesis is correct. So one of the fundamental processes that is contributes to aging phenotypes and multiple chronic disorders is low-grade sterile inflammation associated with fibrosis. Another whole set of processes include macromolecular and organelle dysfunction with issues related to DNA, proteins, mitochondria, et cetera. Stem and progenitor cell dysfunction and cellular senescence, which is what we've focused on. So I think, again, as everybody is aware on the call, cellular senescence is a cell fate. Much like replication, differentiation, or apoptosis, it occurs across the vertebrates. It can occur at any time from conception on, and it appears to be a response to damage-related signals for the most part, including repeated replication. But even things like mechanical stress, shear stress, damage-associated molecular pattern protein and factor detection, pathogens, various drugs can push a cell into senescent cell fate. Unlike other cell fates, senescence takes, at least in culture, 10 days to 6 weeks to become fully established. So it's not like apoptosis, which takes 18 hours, for example. And there are at least 4 transcriptional factor networks that can help to drive a cell into senescence, and some or all of these may be involved in different kinds of senescent cells, but you don't necessarily need all of them to be upregulated. So included are P16 and retinoblastoma protein, the P53, P21 pathway, and a couple of other pathways. But not every senescent cell is increased P16, not every senescent cell is increased P21. They don't have to have this. And as senescence develops, cells lose the ability to divide, essentially irreversibly. And some, but not all senescent cells, anywhere from 30 to 70%, can acquire a senescence-associated secretory phenotype. And there are a couple of interrelated pathways through which this can develop. But it's important to remember that it's only 30 to 70% under most circumstances of senescent cells acquire the SASP. And the SASP may be what is involved in driving tissue dysfunction and diseases related to cellular senescence. Senescent cells also, very much like cancer cells, are resistant to apoptosis. And I'll talk about that a bit more in a moment, but this is the key to developing some of the initial drugs that are senolytic. So senescent cells accumulate in various tissues in humans and other vertebrates with increasing age. This shows in healthy renal transplant donors who are older versus younger. So they're donating a kidney, by definition, they're pretty healthy. They still have increased senescent cell burden. And incidentally, there are no completely specific or sensitive markers of senescent cells. We typically, in clinical trials, measure, we're measuring 48 different things to track these cells. You can devise panels of 6 or 10 or whatever of them that will give you a rough idea of whether a cell is senescent or not, or whether there's evidence of senescent cells in the circulation and so forth. There are no completely sensitive and specific markers. Senescent cells, as I mentioned, accumulate with various disease states, even in very young individuals. So this shows a younger obese diabetic woman with senescent cells, if the SA beta galactosidase is actually targeting senescent cells here, around blood vessels, mainly in her adipose tissue, whereas a slightly older but non-diabetic, non-obese individual does not have these cells near their blood vessels. So in adipose tissue, the senescent cells can include senescent endothelial cells, senescent pericytes, and senescent adipose progenitors. And it looks like even differentiated cells including differentiated fat cells, can acquire senescent-like properties. Small numbers of senescent cells can cause damage. If you transplant them around the knee joint of a mouse, you cause osteoarthritis in that mouse compared to transplanting non-senescent cells. Mice don't get osteoarthritis unless you injure them. You know, it's not like hamsters that get spontaneous osteoarthritis the way that humans do, but this looks for all the world like an X-ray of a human with osteoarthritis. And this is very, very few senescent cells transplanted not into, but around the knee joint. And that's sufficient to cause this state after a couple of months. If you transplant very small numbers of senescent cells into middle-aged animals, you can make them develop an accelerated aging-like state with frailty, early death, and they die early after a lag period of every kind of disease that mice normally die of later. There isn't one particular disease, they die earlier of all diseases. And having 1 in 10,000 cells being a transplanted senescent cell is sufficient in middle-aged mice to cause this to happen. So transplanting 1 million senescent cells is sufficient. If you transplant less than 1 million senescent cells, like 500,000, nothing happens. Whereas if you take that same middle-aged mouse, high-fat feed it, and transplant 500,000 senescent cells, that's sufficient to make the animal develop this accelerated aging-like state. So there's a threshold phenomenon. Above a particular threshold, it appears that disorders can be driven. Below that threshold, it appears that the immune system of the individual is able to clear senescent cells because that's normally how they're removed. Sorry, I'm trying to get to the next slide. I don't know why it's not advancing. So a very important paper by, well, first of all, senescent cells were first described by Hayflick and Moorhead back in '61. It was observed at the NIH that senescent cells increase in tissues of humans with increasing age. And later we did some work showing this in adipose tissue in rats that were double barrier reared and cesarean born and all the rest of it to make sure they're not exposed to external environmental influences. Eugenia Wang noted in 1995 that senescent cells are apoptosis resistant, much like cancer cells. We found that senescent-like preadipocytes in the early 2000s produced TNF-alpha, and this was, you know, Judy Campisi published the SAS back in 2008, but she was talking about it before that, one of the components of which is it can be TNF-alpha. So what really drove us to trying to figure out if we could eliminate senescent cells, what would happen, was a paper by Norm Sharpless, now director of the National Cancer Institute, back in 2004 in JCI, where he showed that if you intervene with things like caloric restriction or the Ames dwarf mutation that extend health span, you delay senescent cell accumulation. So that led us to ask, since there's this association, is there a causal link? And so we started back in 2004 trying to develop senolytic drugs. And we did it by making fusion proteins that would bind on one end to a senescent cell with a toxin on the other, or at least we tried that. We tried high throughput screening, got nowhere. And then finally in May 2013, we came up with a hypothesis-driven approach. And that was, you know, senescent cells resist apoptotic stimuli. They don't die, but they kill the cells around them. So why are they living and how are they able to kill the cells around them, yet they survive this? And the other thing that we used to develop these agents was that in many respects, senescent cells are like cancer cells that don't divide. In fact, there are people who feel that cancers arise from normal cells that go through a phase of senescence, then becomes cancer stem cells arising from the senescent cells, and then a fully blown cancer. At least that's a hypothesis. It's around at the moment. But senescent cells have a partial Warburg shift, and in other ways, they resemble cancer cells, including upregulation of pro-survival pathways. So we use bioinformatics approaches to search for what we call SCAPs, or senescent cell anti-apoptotic pathways. We found 5 of them. Since then, at least 3 more have been discovered, and they appear to interact in a network, such that you know, in many senescent cells, these pathways are redundant. And the pathways, the precise pathways different kinds of senescent cells use to defend themselves against apoptosis vary on the originating cell type, the cause of senescence of species, and a bunch of other things. So not every senescent cell is the same. Not every senescent cell depends on the same SCAP pathways. And in some instances, senescent cells use 2, 3, or 4 pathways to evade apoptosis. So targeting one of them will not kill those senescent cells. We used RNA interference approaches to interrogate this network, and we found that different human kinds of senescent cells depend on different components of the SCAP network to evade killing themselves because of their own SASP. For example, preadipocytes use the dependence receptors, like the ephrin-related receptors. They use pathways that are related to the Src kinase dependence receptor pathways to evade apoptosis. Dependence receptors are receptors that have to be occupied if they're on a cell, or they will kill the cell if they're unoccupied. The one everybody's familiar with is the testosterone receptor in the case of prostatic cancer. So the way, if these receptors are unoccupied, kill cells is through Src kinase mediated mechanisms. And amongst the most important dependence receptors are the ephrins, which keep cells alive if they're in contact with other cells, but if they're not in contact with other cells, they will commit suicide, basically. We found in human endothelial cells, though, they did not depend on these kinds of dependence receptors or Src kinase. They depended a lot more on HIF1 alpha, HSP90, and BCL2 related mechanisms. So we used bioinformatics approaches again to look for drugs, knowing this RNA interference network. And we purposely went after drugs that have already been in the clinic or that are natural products and that have short elimination half-lives to start working with first. We found about 20-odd drugs. We only published a few of them so far using this approach within a very short period of time. We decided to focus initially on a drug, dasatinib, which has been on the market since 2006. Any physician can prescribe it. It's used for tumors that secrete pro-apoptotic factors like B lymphomas, that have something like a SASP. And it works by allowing those factors that those tumors are producing to kill the tumor cell by disabling its defenses. So that's exactly what we are after. It's got a good safety profile. It's about to come off patent. And it's used not only for treating cancers, but scleroderma and a bunch of other things. We predicted it would kill senescent preadipocytes, but not HUVECs, and that was the case. With HUVECs, we went after agents that would target HSP90, BCL2, et cetera, because we knew that preadipocytes would not be killed by those things. And we settled on quercetin to begin with. It's a natural product, again, a short elimination half-life. It's what makes apple peels taste bitter. And then it killed HUVECs, but not preadipocytes as we predicted. So we combined them and we found we could kill both cell types. See if I can make this go. I'm having trouble with this. There. Oh, that's backwards. So that's the path, the network I showed you before. This is what that combination of agents targets on that network. Since then, a number of other agents have been found using this kind of mechanism-based approach. And more recently, it's been possible to generate senolytics using random approaches. So this is exactly the opposite way around of usual drug development. So random high throughput screens have only recently been developed by people like Paul Robbins. And then people have been developing prodrugs and things like that based on some of the characteristics of senescent cells. I'd emphasize that none of these drugs are completely sensitive for senescent cells or completely specific. For example, SA-beta-gal activated toxins, SA-beta-gal is not sensitive and specific for senescent cells. 2/3 of senescent cells express SA-beta-gal, and a lot of activated macrophages that are not senescent also express it. So it's very difficult to necessarily target senescent cells if you go after a single molecular target. I mentioned before that BCL-XL was one of the things that we found protects senescent endothelial cells. So another drug that we tested was navitoclax and other BCL inhibitors. We predicted it would kill senescent endothelial cells, but not preadipocytes by apoptosis, and that was the case. And this came out about the same time as a paper from another group 15 days apart, showing a similar sort of thing. So our view, and this could be wrong, it's just our particular perspective, is that it's better to go after a network of than a single molecular target. So we're not using the classic drug development paradigm of a single target, single drug, single target, single disease. We're going after a network to try to improve specificity and sensitivity, and reduce and flatten side effect profiles. We're going after agents that hit multiple targets or combinations of agents, and we're going after multiple conditions that could be downstream of senescence. So this is a very different approach than the usual drug development approach. One of the agents that we found is senolytic is fisetin, which I'll talk about a bit more. It is it's closely related to quercetin. It's one hydroxyl group different. It's got a shorter half-life, around 3.5 hours or less. We predicted it would kill senescent human preadipsites, but not, HUVECs, but not preadipsites, and that was the case. So when we're trying to develop these drugs, we are mindful of the FDA. All of our trials are FDA approved, and we did things that they would want. So we have to test them in experimental animals, at least 2 species, in humans. And one of the things we do in humans before we go to most trials is take the target tissue, put it in organ culture, and then add the drugs. We found that very brief exposure to senolytic drugs is enough to initiate the apoptosis process, which takes 18 hours to complete. So an exposure of an hour or 2 is all it takes to these drugs. You'll recall that I said it takes 10 days to 6 weeks for new senescent cells to form. Therefore, we use these drugs in a hit and run approach. The FDA did not want us or require us to give pharmacokinetic information, partly because these agents are known and partly because we're using a hit and run approach. We're not trying to get a steady state level. We're not using continuous dosing. We're using single or short bursts of dosing to try to get a high tissue level, not a blood level, and high enough that we can initiate the process of apoptosis. And if the drugs are gone an hour later, we don't care. In fact, we're looking for drugs that'll be gone quickly. We looked on purpose for drugs with short elimination half-lives so we could use this hit and run approach. And then what we showed to the FDA is that these drugs would go on, even after they're removed, to allow senescent cells to die in relevant human tissue explants. And this just shows using TAFs, which is one of the better markers of senescence, that, for example, dasatinib and quercetin do this in adipose tissue samples from obese compared to lean subjects taken from the operating room where we add these agents. And they kill through apoptosis, and we're able to get rid of SAS factors, but not affect things like adipsin that are present in normal adipose tissue. We also did experiments where we transplant senescent cells, as I showed you before, that are labeled or versus non-senescent cells, give the drugs to animals. We predict that 30 to 70% of senescent cells should be killed because that's the percent that have a SAS. These drugs will only kill senescent cells that have a pro-apoptotic SAS. They will not kill what we call helper senescent cells that do not have a SAS. So we're after the senescent cells that are pro-inflammatory and with a tissue-destructive pro-apoptotic protease-producing SAS. We found in those transplanted animals that I showed you before that we're able to alleviate frailty, we're able to restore their remaining lifespan. We found in physical dysfunction in aging mice that we were able to alleviate frailty as well. We're able to extend median more than maximum lifespan with both dasatinib and quercetin and fisetin. And under the same conditions in intact mice where you're removing P16 positive cells, again, some of which are senescent, but there are many cells that have high P16 that are not senescent. Under the same conditions, we didn't find much of an increase in median lifespan. We found a delay in death from all causes that mice die of, the converse of what we found when we transplant senescent cells, hopefully beginning to fulfill cost postulates. And there are many conditions now that these and related agents by various groups who've developed various kinds of senolytics around the world have found can be delayed, prevented, or alleviated in mouse models. So we have clinical trials underway. We have 12 clinical trials underway. There are other groups with many more clinical studies underway around the country and around the world. I'll explain a couple of them, but they're all small, either phase 1 or phase 2A trials, where we try to conduct trials in parallel and where we look at the same parameters across all the trials. So we have a floor of parameters that are related to fundamental aging processes, both at the blood, urine, buccal swab, as well as phenotypic levels. And as was mentioned in the introduction, we have a network, the Translational Geroscience Network, that's funded by the NIH to conduct these trials. And there are other groups who are part of this network for some of the trials, like St. Jude's and the Stedman Clinic. So our strategy, right or wrong, I don't know, this is for discussion, is to go after conditions that are serious, for which there's no really good treatment, where we know that those conditions are related to senescent cell accumulation. And because we're worried about risk-benefit ratios, we don't know what the downsides of senolytic treatment are going to be. So our focus has been on going after these kinds of conditions in parallel rather than in series, and going after things that our IRB and the FDA would not have a problem approving. We've also opted for systemic over local administration because our view is, and this is arguable and different groups have different views on this, local is actually often systemic, except I think for skin application. For example, if you inject these agents into a damaged area, typically that damaged area has an increased blood supply. If you're dealing with membranes and things like that, synovial membranes are broken down. So you're really giving the drug systemically anyway. We found, as I'll talk about in a moment, that senescence spreads from senescent cells to non-senescent cells in an endocrine manner. So we've got evidence that if there are a lot of senescent cells in the knee, for example, you find them in the brain. And this just shows that if you transplant labeled senescent cells into mice, the recipient's own cells start becoming senescent at a great distance. If we transplant them into the peritoneum, we find senescent cells in the limbs of mice that are the recipient's own cells. And there are a variety of mechanisms through which senescence spreads, but it's not just paracrine, it's endocrine. This shows that in obese mice that are insulin resistant, if you treat with, in this case, intermittent, because all our trials we give intermittent administration, dasatinib and quercetin in mice, you can alleviate glucose intolerance. And we show by clamping that it's through decreasing peripheral insulin resistance. Remember, with these drugs, we're targeting not only cells and we're targeting senescent cells in adipose tissue, which most senolytic drugs, with few exceptions, do not. They do not target senescent preadipocytes very well at all, because you have to go after the SARC and dependence receptor pathways. We found that these agents, unlike the situation, for example, in Incotac mice, we do not kill activated macrophages, which have high P16 and high SA beta gal. So these drugs do not kill activated macrophages, but they do kill senescent cells under the same conditions when we take adipose tissue explants and look for macrophages versus adipose progenitors. We find, though, that senescent cells do attract, activate, and anchor immune cells. If you inject labeled monocytes into the tail vein of obese DBDB mice, they accumulate in their visceral fat. If you pre-treat those animals, however, with dasatinib and quercetin, then inject the labeled monocytes, they no longer track to the adipose tissue. So our view is that the decreases in macrophages that we see in adipose tissue, for example, in both our clinical trials and in mice after dasatinib and quercetin, are related to decreased trafficking of macrophages and monocytes to adipose tissue and activation into macrophages. So in a small clinical trial, we looked at a few patients with obesity, diabetes, and renal disease. We did a fat biopsy at day 0. We gave 3 days of dasatinib and quercetin, which are, the combined elimination half-life is 11 hours, so they're completely gone by a day and a half. We looked 11 days after the last dose at day 14, and we found that P16 positive cells and SA beta gal positive cells and repeat adipose biopsies in these subjects were decreased, as were activated macrophages and as were the fibrosis that occurs in adipose tissue related to senescence, where you get formation of crown-like structures. We came up with a composite of blood measures related to the SASP with David Allison at University of Indiana. He helped us devise this. And we found that in this small trial, we were able to reduce this panel of blood biomarkers of senescence-related factors. At the moment, though, as I mentioned, we're measuring more than 40 things in all of our trials. It isn't just this limited panel anymore, including circulating senescent cells. In another small open label trial with all the problems that has, we looked in idiopathic pulmonary fibrosis, a disease which appears to be senescence related, based on work in bleomycin treated mice, and also looking at lung tissue in patients with idiopathic pulmonary fibrosis. A prominent problem that they have, these patients have, is frailty. We knew that it would take a long time to improve the lung issues, but we found that frailty parameters were improved after 9 doses of these drugs over a 3-week period, 5 days after the last dose. But again, this is open label. There are all the problems with that. There could be learning effects. So at the moment, there's a double-blind, placebo-controlled, 3-center trial beginning of these agents for IPF across the Translational Geroscience Network. Others, Miranda Orr and Nick Musi at San Antonio found in mouse models, as problematic as they are, of Alzheimer's-like disease, that these agents appeared to reduce neuroinflammation, decreased tangles, and actually resulted in some brain regrowth. And Mark Mattson found a similar thing at the NIH, working independently from them. So we've got 3 trials underway now in Alzheimer's disease. One of them is a small trial, which is beginning principally in San Antonio. And then there's a larger trial, which the NIH the Alzheimer's Foundation has funded at Mayo to look at target engagement in patients with tau-positive Alzheimer's disease, with comparing a run-in period to a post-treatment period, but mainly looking at CSF senescent cells, because this is target engagement and safety. And there's a third trial which is being funded by the Alzheimer's Drug Discovery Foundation called STOMP-AD, which is a multi-center randomized controlled trial of these agents for Alzheimer's disease. Sandeep Khosla down the hall from me did experiments looking at age-related osteoporosis. We found with dasatinib and quercetin in mice that there's an increase in both cortical and trabecular bone in some instances. Like with any removal of senescent cells, this appears to be due to paracrine effects on stem and progenitor cell dysfunction in bone. So we found that treating with dasatinib and quercetin reduced osteoclast formation because of effects on stem and progenitor cells that give rise to osteoclasts, which resorb bone. And we had the opposite effect on osteoblasts, which make bone. So we found there's both an increase in new bone formation and a decrease in resorption with these drugs. And that's led to a trial which is NIH funded and FDA approved, which is now underway, comparing dasatinib and fisetin, dasatinib and quercetin combination to fisetin to placebo in elderly patients with age-related osteoporosis. And that trial is about halfway through now. With collaborators at St. Jude's, found that in childhood cancer survivors, so these were people who had treatment with alkylating agents or anthracyclines or radiation for leukemias and other childhood cancers before the age of 10, these people develop an accelerated aging-like state, at least a proportion of them, something like 10%, when they're between 35 and 45. And they're developing Alzheimer's disease, they're developing frailty, they develop diabetes, they have osteoporosis. And working with the St. Jude's group, we found that they have senescent cell accumulation as well. These people when they're in their 30s and 40s and have this accelerated aging-like state. they're also dying early. So we have a trial underway at St. Jude now, and again, it's comparing dasatinib and quercetin to fisetin in childhood cancer survivors to ask if we can alleviate frailty in these individuals using Fried criteria. And then we're also obviously looking at safety and tolerability endpoints, as well as senescent cell burden. With Stefan Tullius at Harvard, we've been working on transplantation. Currently in the US, about 35,000 kidneys are thrown away every year from unrelated donors, like in car accidents, because they're over age 50, because we know that these organs don't work very well if they're from older subjects. So with Stefan, we did heart transplants from young mice to young mice or old mice to young mice. And we found that if you transplant hearts from old mice to young mice, you get, much like I showed you before in the transplant models, the organs don't do very well. There's more rejection. There's more mitochondrial DNA released, which contributes to rejection. And there's spread of senescence to the recipient's own tissues at a distance, including their liver. So, and that goes along with the transplant studies I showed you before. So when we treat these mice, either the donors, the organs being transplanted, or the recipients with senolytics, we're able to alleviate these effects. And that's led to 2 trials, which are just beginning, looking at trying to rehabilitate organs before transplantation. In the case of kidneys, we're doing it with the Brigham and Women's in Boston, and asking if we can rehabilitate kidneys from older donors before they're, and then look at their function while they're on perfusion before being transplanted. And we're doing the same thing with liver transplants with the University of Groningen in North Holland. Again, where the organs from older donors can be put onto perfusion devices. They normally are anyway for transportation. And we can actually measure bile production and those kinds of things in the livers and treat or not with senolytics. With Paul Robbins and Laura Niedernhofer at the University of Minnesota, we found that mice infected with a beta coronavirus that is somewhat similar to human coronavirus, although mice don't have ACE2 receptors. We found that old mice all die within 12 days of being infected, young mice don't. If we treat the old mice with senolytics, we're able to delay their death. We're able to reduce cytokine storm, and we're able to get restoration of antibody responses. Similarly, we did a lot of human cell culture and organ studies, and that allowed us to get an emergency authorization to do a trial that's several trials now for COVID. One of them is called COVID fisetin, and that's for people over age 60 or people age 18 to 59 with risk factors for progression of COVID, which incidentally are the same kinds of things that are associated with cellular senescence, things like obesity and diabetes. And in this trial, which is randomized and placebo-controlled, and which is about 1/3 of the way done, we take people who are hospitalized with proven RT-PCR proven coronavirus, and we give them a short course of senolytics, and we're looking at a variety of outcomes, mainly safety outcomes, also target engagement, and to see if we can reduce progression to oxygen requirement. There's a second trial just beginning that's funded by the NIH, by the National Institute on Aging, which is a multi-center trial across the Translational Geroscience Network in people who test positive for coronavirus in nursing homes to see if we can prevent progression to disability and death. So those trials are both with fisetin, because that's the main agent that we used in the preclinical studies. So I'll just finish, sorry for taking up so much time, about regulatory issues. We have not really had big problems with the FDA. We found them to be very supportive and cooperative. I think it's partly because we're not a company, so we're not in an adversarial relationship with them, as it were. Also, our studies are for disease indications, so they're FDA-regulated trials. So no matter what, if we have to we had to go through, we found it a lot easier to take already approved agents like dasatinib into trials than we did with fizetin. Fizetin, it took us 2 years to get to the point where we could do a trial. We had to do a full IND as if it's a new chemical entity. Fizetin is present in strawberries and all kinds of things, but you'd have to get the dose we're giving, you'd have to eat 15 pounds of strawberries in 5 minutes to get the dose we're giving. But the FDA considered it a new drug. So we had to go through a full IND process, 460 pages. Every page is hard as writing an R01 page. We had to do safety and tolerability. We had to go to monkeys. We had to do a lot of the explant studies I showed you, the cell culture studies, the mouse studies. We had to do a lot of mass spec work to prove there are no pesticides, heavy metals, or solvents. We had to have the farms inspected where the trees came from, that we got it from. We had to set up GMP manufacturing. So it is really, really hard to take even a food product like fizetin, which everybody is eating all the time, to use it as a drug. Nevertheless, even though it was a lot of work, they allowed us to do accelerated toxicity and other things. And as I mentioned, we were not required to do pharmacokinetics because we're using these drugs in a hit and run manner. We had to also write investigators brochure. We have to do our own stability testing. We had to establish a drug depot, everything just like it's a new chemical entity. So that was a real education. And we've also found, approaching the FDA about senescence-associated diseases and looking at them in parallel, it seemed to be an easier approach for us than going after giving these agents to otherwise healthy individuals with aging as an indication, as our starting point. So that's just the way that we happen to do things. I don't know that it's the right way, but it's what we're doing with other senolytics that we've got in the development pipeline. So I'll just conclude by saying the target of senolytics is senescent cells. It's not a single molecular pathway. We've gotten away from the one drug, one target, one disease paradigm. We found that a lot of the way that they work is by alleviating progenitor and stem cell dysfunction. I didn't have time to go into that, and decreasing inflammation. Intermittent treatment looks like it's as or more effective than continuous treatment. We had to show that to the FDA, and it actually reduces the opportunity for side effects considerably. We're able to alleviate multiple chronic diseases, enhance health span and lifespan in mice, but that doesn't mean it's going to be true in humans. So our view is these drugs should absolutely not be taken by people outside the context of clinical trials, in the way at least that we're giving them. You know, we don't know what the downsides are going to be. And the main thing we're looking for in all of our studies is downsides. The main thing our DSMBs are looking for is, what are the side effects? So we look like crazy for side effects for these agents. And I lie awake at night, every night, worried about this. So I'll just conclude by thanking a lot of people, many of whom are on the call probably. The NIH has been unbelievably supportive. There have been individuals who've helped us out. We're also grateful to the Alzheimer's Association, Alzheimer's Drug Discovery Foundation, and the American Federation for Aging Research for all of their help. So sorry for taking up so much time, but you know, probably our way of thinking is a bit crazy. It's a bit different than the others have maybe on the call. And this is, you know, how we've done things. I'm not saying it's the right way of doing things. It's just sort of what our thinking process has been.
Fantastic. Thank you so, so much. I hope you don't mind that. Oh, you already stopped sharing your screen. Thank you so much. That was a lot of information to digest. And I've already have a lot of questions queued up, if you don't mind taking a few of them.
Sure, but you know, my answers are wild guesses. You know, we're only just scratching the surface of this, and we could be very, very wrong anywhere along the way.
I think likewise for everyone else who's questioning. So perhaps Tom, you'd like to go first?
Sure, Jim, great presentation. I guess my question is, are there things, you know, thinking about this from the perspective not only of your own work, but the field as a whole, both on the fundamental side and on the translational side, are there things that you would like to see the government, and by that I mean principally NIH, FNIH and FDA to do differently than they are currently doing?
Well, we've tended to work within the guidelines and frameworks that they have established, so we've looked for a path of sort of least resistance, as it were, even though it's very difficult. They've been incredibly helpful. And I've got to say, the FDA has been very helpful. A lot of the time when you're waiting for a study may proceed letter, they say you're going to wait 60 to 90 days and stuff like that. Certainly in the case of the COVID stuff, they've gotten back to us in 3 or 5 days. When they've had reviews, like panels set up in the FDA, they've given us helpful advice. And one of the things they did was they gave us a study may proceed letter that was about 5 pages long, but it said study may proceed. Could you consider these things as advice when you're doing your trial? And they gave us 5 pages of advice about statistics and other approaches that were unbelievably helpful. And they didn't stop us. They told us, consider these things, and they're drawing on all kinds of other people's experience, and they gave us the benefit of that. So I really, I've been overwhelmed by how helpful they've been. There have been times when at the very beginning, it took them a long time to get back to us, but I've got to say that's where the NIH and I think behind the scenes stepped in and talked to a few people at the FDA and asked them to turn the wheels a bit faster on the COVID studies. And I don't know exactly what happened. I'm only suspecting it, but I think the NIH had a discussion with the FDA and things happened fast. So I just don't have a lot of problems. And the Aging Institute has been just incredible. The one thing I think is the Aging Institute, really should get more money now. And, what we can't do is rob Peter to pay Paul. I think the drug development pipeline starts with basic research, and that's the most important thing by far. And what I don't think we can do is take away from basic research to start paying for translational stuff. So I think now that these agents are moving from the bench to the bedside for the first time, beginning with Nir's work with metformin and Matt and others work with the rapalogs and so forth. Now that they're beginning to move from bench to bedside, there needs to be a big translational push, and that will require some funding. But I don't think that should be done by re-budgeting within the NIA. I think new money has to come in to pay for the translational component, because it would just be criminal, in my view, to take away from the basic science side to support early phase translation.
Just one comment, and that is, I think one issue is getting the Congress to recognize the importance of geroscience, because the NIA top line has been going up, but that has been primarily targeted for Alzheimer's. So I think that their NIA is under some resource constraints in terms of their ability to support additional translational research,
right?
Given the congressional directive that they've gotten. And I think that stems from the fact that no one shows up at the Congress and says, hey, I'm getting older, can you do something about that? They say, my wife got Alzheimer's.
But if you relate Alzheimer's to these fundamental processes and say that, aging is potentially a root cause contributor, and I think you can almost say that now. I think the geroscience hypothesis has been around for 15 years or something. I think it's pretty well beyond a doubt now that some of these fundamental aging processes, you could say with a straight face, are root cause contributors. And especially with Alzheimer's, 80% of the risk can be predicted by your age alone.
Thank you. I think you.
Yeah, can I show a slide, Alison? Can you release me?
Now I'm in fear.
Can you release the slide? No, not at all. First of all, I just want you to know that Jim and I always do this little show. We know each other for so long. I've been admirer of his work. You see why, and he has problems with me, but we're okay.
Well, I always say I've heard many great things about Nir, and some of them are true.
And I say I heard only good things on you. That's my answer usually. So, but I want to just and for Tom, really, for for really the history. Jim and some of us has this effort that started basically 10 years ago or maybe more to get the geriatrician and the translational people with the biologies and try to think of interventions. And I got this program that paid for, in part, for a meeting in Spain in a parador. It's a Toledo parador, but it's not in Toledo, the city. It's like 300 kilometers from Toledo, the city. And we called it a French jail because it was very isolated, but in Spain, but the food was excellent. And that's where we came with those two concepts, one of which Jim is leading and one in which I was leading. So the first one is TAME, right? To show that we can give treatment and prevent age and age-related disease and all that. And Jim was looking at this paradigm, which is to interfere. Then we were talking about interfering in like people before surgery, elderly before hip surgery. Maybe we can give them senolytics and we'll be able to recover them better. And that's where it started running. And then Jim's research on senolytics kind of accelerated not only this paradigm, but paradigm that's related to other age-related diseases. And so those are still kind of the two pathways that we have to push. And as you saw last week with the FDA, and this week, we can work with the FDA, but we need the wave to be bigger. We did go to the Congress, we did go to the Senate, we presented that, and it was fine, but this didn't create the wave that we wanted to. So I just wanted to have the historical note and where did we start and where are we going?
Right. It was good.
Thank you.
Jim.
Thank you for that very eloquent. Oh, thank you, Nir. Carl, would you like to chime in with your question?
Okay, can you hear me? My internet cut out a minute while you were talking. So Jim, don't worry, I don't think most people think you're crazy. Actually, I think that your research program with these small little trials is actually super, super important because, and I think most people in the field think it's important because, there could be a very quick win here and it would be terrible if humanity did not actually go and check it. Plus, we're gonna learn a bunch of important things. So I wanna ask you about biomarkers. The two parts of the question are you specifically in any of the trials looking at aging clocks, methylome clocks or the like at baseline and at post-treatment for at least some subset of the patients? And then separately, sort of what, we don't really have a sort of aging clock equivalent specifically for senescence cell burden. At the moment, it's hard to inspect, we have beta-gal and a bunch of other things that are sort of easy to do with mice or in biopsy, but we don't really have a good sort of senescence clock yet, but there's things coming down the road and I'm curious what you're excited about, if anything, and what best approximations you're using.
Yeah, we measure, we're working with Morgan Levine as part of the analysis facility. So we have something called a facility for gyroscience analysis, where blood samples, tissue, buccal swabs, PBMCs, CSF, urine cells, etc., from all the trials are analyzed centrally across the network. And so in addition to the measuring a bunch of blood tests and urine tests and things in CSF when we get it, we are looking at nucleotides like mitochondrial DNA as divided by total DNA, for example, in the blood, and various kinds of microRNAs. We're looking at methylation clock with respect to the buccal swabs and PBMCs. And as you say, Morgan and others are pretty far down the road for developing ones related to senescence. But we're not just looking at senescence across these trials, because the trials aren't just of senolytics. The R24 that Nir mentioned to is what led to the R33, which exists now. And we're looking at you know, everything we can measure, including CD38 and NAD and NMN. You know, we're looking at you know, everything we can. You know, things related to mTOR activity. You know, so we're trying to look at all the pillars of aging that we can across all the tissues that we can get. We're trying to do it in a standardized way across the trials.
Thank you, Ravi.
Yeah, I had a question. You're leading the AFFIRM Senescence study, which you hadn't talked about. Do you have any early results from that that you could discuss?
The only thing I can say, you know, because it's a double-blind trial, is that we haven't seen, and I'm touching wood, any severe or serious side effects so far. So, you know, that's all that I know, basically.
And once you've done all that work for the IND, effectively the IND for Fisetin, is that something other researchers can now leverage if they want to use Fisetin in a study?
Yeah, and we've done that. So we're working with groups, not just in the US, but in Europe and the UK and so forth, who are trying to deal with their regulators as well, and to try to get some of these agents approved by the EU and the British authorities.
So can I sneak in one more question, which is what you just mentioned about your central facility for testing all of these biomarkers? Is that comprehensive data going to be made available as well? Like
Yeah, Steve Kraszewski is in charge of developing the sort of, and George Kuchel, they're in charge of developing the data repositories and analysis side of it. The actual facility is based at Mayo. Tamara Ciccone is head of it. But the data are analyzed by Steve's group at Wake Forest, and then George Kuchel at University of Connecticut is leading efforts to try to develop ways that we can biobank things, and Steve is developing ways to try to share data once they're available.
Awesome, thank you.
Lovely. Next one up, we have Joe.
Hey there. I saw a talk at BAM last year by Nathan Basisti, who's talking about doing this really big, sort of more unbiased proteomic study on senescent cells, and I haven't followed it since then. But the theme I got, and listening to you today, this was brought up again, is that there's such a diverse phenotype. Different ways of detecting them, different signals, suggesting that maybe there's just some like a few different categories of like fundamentally different biology of things that we're calling quote unquote senescent cells that you know share this basic idea of maybe cell cycle arrest. Is there sort of like a taxonomy emerging at the top level for senescent cells now with different categories?
Yeah, it's beginning. We call them H and D senescent cells, you know, for helper and deleterious, you know, whether they have a SAS but it's deleterious or not. But depends on the starting cell type. As I showed you before, different kinds of senescent cells depend on different pathways for their survival, produce different sets of SAS components. And not only does it depend on the starting cell type, it depends on how senescence was induced and the amount of time since it was induced. So John Sedevy showed, for example, that after a period of time, senescent cells start having transpositional events. You know, it's not, initially they don't, but after 2, 3, 4 months with some human cell types, they start having line 1 transpositional events. So they've got all kinds of chromosomal rearrangements that begin. So they change in structure and function very dramatically after that. We don't know how long they persist in humans, but in mice, it seems in healthy mice, senescent cells generally can persist up to a couple of months, and then they're cleared by the immune system or sometimes before that. So that also indicates that their repertoire of signals that are related to the immune system change over time. And then once they've been around for a while, though, they become resistant to moving, very much like cancer cells. They start expressing don't eat me signals. They you know, have a more exuberant SAS, so they're producing things like proteases that cleave off fast ligand and disable the immune system. They produce IL-6, which inhibits macrophage diapedesis. They produce After a while, not at the beginning, but once they're mature senescent cells, after a few weeks or months, they produce a lot of fibrous tissue around them. And that shields them from the immune system, but also causes scarring. So they change over time as well. And the kind of inducer is very important. And whether there's P16 upregulation versus P53, P21, they're very different. You know, the P21 cells are much more likely to be related to metabolic disease like diabetes. The P16 are more related to passage of time and sort of aging related kinds of processes. They're different, you know, P53, P21 are more upregulated if radiation is induced senescence. So, you know, it's just the same as saying a proliferating cell is not a proliferating cell. They're all different. It's a cell fate. And I think there are many, and I think also that non-dividing cells can become senescent-like. You know, Gina Ellison in London and Joao Passos here showed that with respect to cardiomyocytes. It's looking like it's true for adipocytes and so forth. There's debate about that. Looking like it may be true for neurons too. There may be senescent non-dividing cells.
Thank you.
Thank you so much. I know we're now 2 minutes over time. I would have 2 more questions in the queue in case you have more time, but also totally fine if you have to hop off.
I've actually got to hop off because I'm on the coronavirus task force here, and we review all the patients twice a day, and they've already started for the afternoon. So you can imagine it's a mess. So.
Oh my God. Okay. Well, thank you for doing that. And thank you so much for joining us here today. I know that people were, yeah, it was a really fantastic presentation. I'll be following up with you in case you're able to share the slides. Thank you from everyone here for joining us today. It was really fantastic. Thank you.
Yeah, and thanks. And take everything I said with a grain of salt. All of this changes from day to day. I mean, it literally is very fast moving. And I could be very wrong about some of the things I said very quickly. You know, it's really hard to keep ahead of developments.
Thank you so much. All right,
Thanks.
we let you go. Bye.
Bye.
Thank you so much. Bye-bye.